Hi everyone and welcome to today's lecture on nucleotide metabolism. And as you know, nucleotides are my babies. Love that DNA and RNA as well as, don't forget, ATP. So before we go into the metabolism of nucleotides, which, as you know, metabolism means building up and breaking down, all of the reactions. Before we get into that, I want to remind you of the structure and function of nucleotides. So the first question that I ask you is what exactly are they used in? And if you think of what the examples of the nucleotides are, such as, or I should say nucleic acids as well, if you think of DNA, RNA, and ATP as the three main nucleic acids, that'll help you answer what nucleotides are used in. So when you think of DNA and RNA, that shows us that one of the uses is genetic information storage and transfer. But additionally, we also have ATP as one of the nucleotides. So that means that nucleotides are involved in energy conversion, signal transduction, and coenzyme dependent reactions. And we'll go more over this in a moment. Now, just a reminder of what they actually consist of. So a nucleotide you see over here has the nucleotide base. So a lot of times you hear that as the nitrogen base. It will have the ribose or deoxyribose sugar, and then it will have the phosphoryl groups over here. Okay, and then that's what gets linked together to build the ultimate, 5' 3' chains of nucleic acids. Now, when we talk about nucleotides, I want you to know purine examples versus pyrimidines. And the trick that I always tell students is to think of cut pie. So, like you're cutting a pie, C, U, and T are the examples of pyrimidines, whereas A and G, being adenine and guanine, are the examples of purines. Now, as I mentioned a moment ago, a lot of people forget that ATP is one of the examples of nucleotides or nucleic acids. And so the first thing I want to point out is this question over here, circle star highlight the comparison of ATP to other nucleotides, because I want you to remember that ATP is the most abundant intracellular free nucleotide. Okay, and notice I say free nucleotide because DNA and RNA, for instance, those are nucleic acids made up of linked nucleotides that are not free floating. The free nucleotides from the RNA and DNA, those would be things like DATP, DTTP, the nitrogenous base individual nucleotides. Now, when we talk about the roles of ATP, this relates to what we just said a moment ago. ATP is actually involved in all four roles of nucleotides that I mentioned in the previous slide. So energy conversion, signal transduction, coenzyme reactions, as well as storage and transfer of genetic information. Now, as we start to get into the metabolism of these nucleotides and nucleic acids, I want to introduce you to what's called the nucleotide salvage pathway. And if you are salvaging something, you're basically recycling it, okay? You're finding a way to utilize something that was something else before. So the purpose of the nucleotide salvage pathway is going to be to resynthesize the nucleotide monophosphates from free bases. Now, this is a valuable process basically. I'm not going to make you memorize the details of the figures that you see here. I just want you to know that this pathway exists and that it is allowing us to obtain nucleotides from other previous free bases. And that's very valuable and beneficial because that means that the energy investment of getting those nucleotides is not going to be as high as what's called the de novo pathway, which would be building those nucleotides from scratch. Okay, so recycling old free nucleotides in the nucleotide salvage pathway is beneficial to save energy, to conserve resources. Okay, so now we're going to look at purine and pyrimidine biosynthesis. And so I want you to look at this figure over here. This first half is purine biosynthesis. The second half is pyrimidine. So again, cut pi helps you remember that pyrimidine biosynthesis will lead to C, U, and T, whereas purine leads to A and G. Now, you'll notice the difference over here right at the beginning is that purines are synthesized directly onto the ribose sugar called PRPP. So you see the purine getting synthesized where you have the attachment directly onto that sugar, okay, the base directly onto the ribose. Now, this then leads to the intermediate of IMP, which will ultimately yield A and G. Now, pyrimidine biosynthesis, you notice you first have to link together the I always mispronounce that, carbamoyl phosphate and the aspartate, and that will then form what you see here, the orotate. So this is orotate, and then that gets linked to the PRPP to generate OMP. Okay, so for purine biosynthesis, you see IMP as the intermediate. And then for pyrimidine, whenever you see OMP, that's the pyrimidine intermediate that then leads to our three pyrimidines. And we'll go over later some of the reactions that are ultimately leading to those final nucleotides. Now, you noticed in those previous slides, we were saying AMP, GMP, and so forth, the MP being monophosphates. So right here, you see there's only one phosphoryl group on AMP and on GMP. But when you want to build DNA and RNA, you know that you need triphosphates, right? ATP, GTP. Okay, you need the triphosphate because breaking that bond, breaking the triphosphate bond is going to allow you to create the 5' 3' backbone of those structures. So if you only had the one phosphate, you're not going to be able to link together all the nucleotides to build the DNA and RNA. Okay, so what has to happen once you have had biosynthesis create AMP and GMP in the figures that we just saw on the previous slides, you then need to convert those. You need them to be phosphorylated to generate the nucleoside triphosphates that ultimately allow for RNA and DNA synthesis. Okay, and basically you tend to have an enzyme called nucleoside diphosphate kinase, which you see over here. And when you have kinases, they'll be involved in the phosphate transfers and the phosphorylation to ultimately lead to the triphosphate form of those nucleotides. Now, as we've mentioned so many times in this class, metabolism is not just one-sided. If you have biosynthesis and you have synthesis, you need the opposite to occur as well. You need degradation. So the first thing we're going to look at here is purine degradation. And with purine degradation, I want you to circle, star, highlight the question here of what is the product. So the product of purine degradation you're going to see over here is uric acid. And that uric acid is basically either excreted directly or it gets further metabolized to nitrogen waste products. And your nitrogen waste products depends on what kind of organism you are. So mammals like us excrete urea. And you'll notice that that's right smack in the middle in terms of the water needed and the energy needed. So that's a nice balanced waste product for us. If, however, we were birds and reptiles, then we would be able to excrete the uric acid directly as uric acid. There would be no further metabolism or conversion necessary. But as you see, that's a very energy intensive process. So we're not going to want to do that. Let's leave that for birds and reptiles. The last one that you see over here, or first one, if you're looking at it from left to right, is ammonia. So ammonia is going to be the nitrogen waste product of fish. And the reason that fish can release ammonia as their waste product in terms of purine degradation is because ammonia would be highly toxic. You know, if we have a ton of ammonia, that's going to be a problem for us. But fish, what you notice, they're in an area with very high water available to them. So they're able to utilize ammonia as a waste product because there's plenty of water for excretion to cancel out the highly toxic nature of the ammonia. Okay, so for ammonia, write that this would be fish, use this as their nitrogen waste product. Urea is mammals, such as humans. And then uric acid is used by birds and reptiles. So now, speaking of the purine degradation pathway and purines, that brings us to an ailment that is close to my heart. So gout, the reason this is close to my heart is because my Nana had always suffered from gout, and thanks to medication that we're going to go over in a minute, she was then able to eat some of her favorite foods again, as you can see in that picture. Now, before we get into the food restrictions and all of that, I first want to tell you what exactly gout is. So, gout is a defect in the purine degradation pathway. Okay, basically, you have a problem at this stage in the purine degradation pathway. So, you notice you're degrading the ribose 5P down into the PRPP and the IMP, those intermediates that we talked about when we were building the nucleotides, the purines. So now we're breaking down the ribose into those earlier intermediates and earlier components of the pathway. So now, in gout, you have a problem with PRPP synthase, okay? Synthetase, sorry. And you end up having an increase in this synthetase. So you have a defect where you have too much of this synthetase. And what that means is you are going to go through this pathway overabundantly. Okay, so an increase in the PRPP synthetase leads to an increase in PRPP, an increase in and overproduction of IMP, and ultimately, you end up all the way down the pathway because you're getting more of the reactants that lead to more of the products, more of that product, more of that product. You end up with a buildup of uric acid crystals, and they build up in the joints and the kidneys, and it could be very, very painful. And one of the predominant ones that it's characterized by is the big toe. A lot of people experience it with a buildup of the crystals of uric acid in the joints of their big toe, and you see inflammation and a whole lot of pain, okay? Now, luckily they came out with a drug called allopurinol, circle star highlight. Allopurinol is the treatment for gout because what it does is it inhibits this final enzyme in the pathway. So it inhibits xanthine oxidase, which reduces the production of uric acid. So you don't end up with that accumulation of uric acid. And so you'll notice that people who have issues with gout, like my grandmother had, when they have issues with gout, before they even find out about the medication, one of the things that they are told to do is reduce eating or reduce in their diet things that would have a lot of purines in them. And so one of the big things is reduction in the amount of seafood that you eat, especially shrimps, crabs, lobsters. And so for some people that was brutal to tell them you can't have those foods anymore. But thankfully with allopurinol, they're then able to start eating those foods again, still hopefully in moderation, because the allopurinol is blocking the final production of uric acid, and it's decreasing the rate. It does not completely cancel out all of it, but it decreases the rate of production. Okay, another disease or syndrome that we see from problems in the purine degradation pathway is called Lesch-Nyman syndrome. And this one is really, really, really sad. It's very rare, so you don't see or hear of it as much as you do something like gout. But basically, this one, the problem is over here in this part of the pathway in the enzyme HGPRT. Okay, so these people suffering from Leishman, Nyhan, sorry, I keep mispronouncing that, Nyhan syndrome, they have a deficiency in the HGPRT enzyme. What that means is that they're not properly recycling or salvaging hypoxanthine and guanine bases back up into the cycle. Okay, so you notice you're supposed to have some salvage or recycling here where some of that hypoxanthine is getting recycled back into the higher portions of the cycle. So now you don't have that occurring because there's the deficiency in HGPRT, and so now you have an accumulation of hypoxanthine. If you have an accumulation of hypoxanthine, where is it going to go? Well, it's going to get converted to uric acid. Okay, it's going to get turned into uric acid from xanthine oxidase over here. So when you get an accumulation here, it feeds more into the final product. So you have an overproduction of uric acid, but unlike gout, this is not going to be an accumulation in the joints and kidneys. Instead, this is occurring in the nervous system. You end up seeing really, really unusual neurological symptoms, which you see in the figure over here, that include things like severe anxiety and self-mutilation. And so the self-mutilation is that people with this syndrome will actually bite off their own fingers, bite off their own skin. And you see here, they not only target their hands and their fingers, but they even bite off their own lips and their own mouth portions. They destroy their own teeth and even tongue sometimes. It's a very, very brutal um, syndrome, and it seems to be from an X-linked recessive genetic mutation that leads to a deficiency in that enzyme and then the neurological issues that you see here. The last one of the syndromes or deficiencies that I want to mention in terms of purine degradation is adenosine deaminase deficiency, ADA. And with this one, this is going to be caused by defects in adenosine deaminase. And so you don't have the conversion of adenosine into inosine, which would then lead to the hypoxanthine that we talked about a moment ago and the uric acid that we talked about a moment ago. And so with this one, you end up, not having enough of that inosine getting cycled into the pathway. And with this, when you have the defect in adenosine deaminase, they found that it leads to what's called severe combined immunodeficiency disease, which is written as SCID for short. And that basically means that you end up with a very defective immune system, and you're going to end up having low levels of functional B cells and T cells, and the only way to then treat that is through bone marrow transplant. Although they're also experimentally working on treatment with stem cells as well. So now we're going to switch over away from the purines and onto the pyrimidines. Now, back when we talked about pyrimidine versus purine synthesis, we saw that with the purines, you directly put the the base onto the PRPP sugar. In pyrimidine biosynthesis, you remember you first had to link together aspartate and carbamoyl phosphate. So that means that when I ask where are the 6 atoms of pyrimidine derived from, the answer is that aspartate and carbamoyl phosphate that we saw in pyrimidine biosynthesis. Now, when we talked about biosynthesis of pyrimidines, you'll remember that once we had put together that aspartate and carbamoyl phosphate and then linked that to the sugar, you then ended up leading to UMP as the main product. What then has to happen for us to ultimately get CTP from that UMP intermediate or original product is that you have to have first the UMP gets converted to UTP through basically a sequential phosphorylation because you have one phosphoryl group originally, then you have to add a second one, then you add the third one. So at this point, using kinases in order to phosphorylate that UMP twice into UTP, you then end up having to aminate that by what's called CTP synthetase. Okay, so you're aminating that, which you see the amine group right over here highlighted in blue, that NH2 that's been added to it, and that ultimately generates CTP. Okay, so first in biosynthesis, you get to make the UMP that then gets phosphorylated and phosphorylated again by kinases. So you have UTP, which is triphosphate, 3 phosphoryl groups here, and then you aminate that, see that amine group over there, to make CTP. Now, like I keep emphasizing, whenever you talk about metabolism, there's 2 sides of the story. So you also have to take a look at pyrimidine degradation. Now, the final products you can circle star highlight over here, they are beta alanine and beta aminoisobutyrate. Okay, beta alanine is basically used in the synthesis of acetyl CoA, which we're going to see talked about in some of our other lectures, how valuable acetyl CoA is. And then beta aminoisobutyrate, we see that in valine degradation, which may pop up in one of the other lessons. Now, what I want to focus on in this slide when we talk about pyrimidine degradation, which you see here, degrading those pyrimidines into those 2 final products, this relates to something very important in the medical field, which is highlighted in this figure here. There's a drug called 5-fluorouracil, okay? And when you put these modified nucleotides into the body, cells that are basically high in terms of their replication and their DNA synthesis and RNA synthesis, so in this case uracil for RNA synthesis, when they are highly replicating cells, these modified pyrimidines can basically impact their ability to survive. And so when I say highly replicating cells, that tells us cancer cells, tumor cells. So 5-fluorouracil is basically used in the treatment of cancer. Now, when they give a patient 5-fluorouracil, they give them a much higher dose than you would expect because in the body there's an enzyme, which let me circle it over here. So in this pyrimidine degradation pathway, you see this enzyme, dihydropyrimidine dehydrogenase. Okay, so that would normally be part of breaking down the uracil. Okay, if you're giving the patient 5-fluorouracil, this enzyme in a normally functioning body with the proper amount of this enzyme, that's going to degrade a large percentage of the 5-fluorouracil that you're giving to your patient. So they prescribe that treatment in a much higher dose to compensate for the fact that 80% of that 5-fluorouracil is going to get degraded in the healthy body. Now, in some patients, it turns out that they have a dihydropyrimidine dehydrogenase deficiency. They're deficient in this enzyme, and you might not have noticed it otherwise, but if you give them that medication of 5-fluorouracil, what's going to happen? They don't have the proper amount of that enzyme to break it down. So now instead of 80% of that heavy dose of 5-fluorouracil getting degraded to bring it to the proper amount, instead the patient is getting 80% of the dose. Okay, so they end up having tissue toxicity, and that becomes very dangerous for them. Okay, so that's an example of how having a defect in the pyrimidine degradation pathway can be very problematic in a medical example. So it creates a tissue toxicity effect for a drug that's supposed to help save the person during their cancer struggle. Okay, now I just want to point out in all of this that there's also deoxyribonucleotide metabolism. And all I want you to know for this one is that it relies on an enzyme called ribonucleotide reductase. And that ribonucleotide reductase requires our good old buddy NADPH, which is going to supply 2 electrons to help that enzyme function. So now, if you recapped what we have created so far, we've looked at A and G synthesis. Those are our purines. And out of the pyrimidines, we looked at U and at C. But the only one that we haven't gotten to yet is T, so thymine. And so that brings us to thymidylate synthase. This is ultimately going to be responsible for catalyzing a methylation reaction that converts the UMP to TMP. Okay, so the conversion of UMP to TMP, that's thymidylate synthase here. Okay, now once you have that, keep in mind that you also then need to have kinases to sequentially phosphorylate that into TTP because again, we need the triphosphate form of these bases in order to create our DNA. Okay, so when you look at thymidylate synthase, that is going to be in terms of getting us TTP and ultimately thymine. Now, there are some inhibitors of that enzyme, thymidylate synthase, and if you use one of these inhibitors, you'll then be able to block the activity of that enzyme, which ultimately blocks the conversion or creation of thymine, well, first thymidylate and then thymine, which is valuable to us as potent anti-cancer medications. So raltetrexate and methotrexate, those are both potent anti-cancer medications. So circle star highlight that they are potent anti-cancer medications. And because they are inhibiting thymidylate synthase, they are going to inhibit that DTMP synthesis or TTP synthesis, and that ends up blocking DNA replication in the rapidly dividing cancer cells. And sometimes they'll combine one of those, such as raltetrexate or methotrexate. They'll combine that with something like the 5-fluorouracil that we mentioned or something called verapamil, and they'll basically create an anti-drug cocktail to help kill the cancer cells before drug resistance begin. So they'll try and target multiple of the pathways that we've mentioned so far. That then brings us to the review questions. Again, please make sure to always send me your answers to these review questions right away as soon as you get to them in this video. Send them to me in the Remind app. If you have any trouble with that, you can send me an email to let me know that you're having issues with your Remind app. Okay? Again, please make sure to send them anytime, day or night. And when you send them, please send a picture of the slide that you are answering. That helps me to know right away. If you just send a letter, it's going to be difficult for me to know whether you are correct or not because I won't know exactly which question you are answering. Another review question for you to send in the Remind app. Again, please send them right away. And that is it for today. As always, if you have any questions or comments, please send me a message anytime, day or night. And always, if you can please send me a picture of the exact slide that you are asking a question about, that helps me to really make sure that I know that I'm answering you correctly. Thank you and have a great day.